Bismillahirrahmanirrahim. We will study the metabolism of triacylglycerol and compound lipids. In this chapter, we will study the biosynthesis of triacylglycerol and compound lipids with their some clinical significances. As we know that lipid constitutes about 15 to 20% of the total body lipids, of which triacylglycerol comprises about 80 to 95% of the total body lipids. This triacylglycerol is stored in adipose tissues, and these adipose tissues serve as the stores of energy. This triacylglycerol is a non-polar molecule which is hydrophobic in nature. Now we see the synthesis of triacylglycerol. Triacylglycerol is an ester which is derived from the glycerol and 3 fatty acids. That is, in glycerol molecule, the 3 fatty acids are esterified with the glycerol molecule, of which the fatty acid which is esterified at carbon 1 of the glycerol is always unsaturated fatty acid, while the fatty acid which is attached or esterified to the second carbon is typically the unsaturated fatty acid. The fatty acid at the third position of the third carbon of the glycerol may either be saturated or unsaturated. Now we see that as the triacylglycerol contain a glycerol molecule, the glycerol molecule is a 3 carbon containing molecule which have 3 hydroxyl groups. These hydroxyl groups are responsible for the solubility of this molecule in water. So due to the presence of these hydroxyl groups, the glycerol molecule is hydrophilic in nature. This glycerol molecule is a central component of many lipids, including triacylglycerol and phospholipids. Now, for the synthesis of triacylglycerol, 2 molecules are required. One is glycerol in the form of glycerol 3-phosphate, and the other are the fatty acids in the form of Acyl-CoA. Both these forms are the activated forms of glycerol and fatty acids. Most of the synthesis of triacylglycerol takes place in liver and adipose tissues, and to some extent in other tissues as well. At the cellular level, the synthesis of triacylglycerol takes place in endoplasmic reticulum, because most of the enzymes required for the synthesis of triacylglycerol are present in endoplasmic reticulum. The major enzyme for the synthesis of this triacylglycerol is the transferases. For example, the glycerol 3-phosphate acyl transferase. Some of these synthesis of triacylglycerol takes place in mitochondria as well. Now we see how the synthesis of glycerol phosphate takes place. In the synthesis of glycerol, there are 2 mechanisms for the biosynthesis of glycerol 3-phosphate. That is, one from the glycerol and the other from the dihydroxyacetone phosphate. This glycerol which reaches the liver is directly converted to glycerol 3-phosphate by the enzyme glycerol kinase. This enzyme glycerol kinase phosphorylates the glycerol into glycerol 3-phosphate. The other pathway is the synthesis of glycerol 3-phosphate from dihydroxyacetone phosphate. This dihydroxyacetone phosphate is the intermediate in the glycolytic pathway of glucose. This dihydroxyacetone phosphate is converted to glycerol 3-phosphate by the action of enzyme glycerol 3-phosphate dehydrogenase. This enzyme glycerol 3-phosphate dehydrogenase converts the dihydroxyacetone phosphate into glycerol 3-phosphate in the liver. This enzyme reduces the dihydroxyacetone phosphate into the glycerol. Some of the synthesis of this triglycerol 3-phosphate takes place in other tissues as well, like kidneys. This diagram shows the synthesis of glycerol 3-phosphate in the liver and adipose tissues. From the diagram, it is clear that both the pathways of synthesis of glycerol 3-phosphate, that is either from the glycerol with the action of glycerol kinase or from the dihydroxyacetone phosphate with the action of glycerol 3-phosphate dehydrogenase, both these pathways are present in the liver, while in adipose tissues and only the single pathway is present in adipose tissues, that is from the dihydroxyacetone phosphate. The reason is that the enzyme which is phosphorylates the glycerol to glycerol 3-phosphate, that is the glycerol kinase, is absent in adipose tissues. These are the reactions which show the synthesis of glycerol phosphate from the glycerol and dihydroxyacetone phosphate. This glycerol, we see that the glycerol is converted to glycerol 3-phosphate and the reaction is catalyzed by glycerol kinase, and the phosphates are provided by the ATP, which after giving phosphate group, phosphoryl group to the glycerol is converted to ADP. On the other hand, this dihydroxyacetone phosphate is converted to glycerol 3-phosphate by the action of glycerol 3-phosphate dehydrogenase. In this reaction, we see that the keto group of the dihydroxyacetone phosphate is reduced to hydroxyl group, and the reducing equivalents are provided by the coenzyme NADH. So in this way, the glycerol 3-phosphate is synthesized from glycerol and dihydroxyacetone phosphate. This diagram shows that the glycerol 3-phosphate and dihydroxyacetone phosphate are interconvertible molecules, and from the glycerol 3-phosphate, phosphatidic acid is synthesized, and from this phosphatidic acid or phosphatidate, glycerol is formed, diacylglycerol is synthesized, and then this diacylglycerol is converted to triacylglycerol and the phosphatidylcholine and phosphatidylethanol. From the phosphatidic acid, the other compounds which are synthesized are the cardiolipin and phosphatidylinositol, and then this phosphatidylinositol is further phosphorylated into phosphatidylinositol 4,5 bisphosphate. The lipids which are synthesized from dihydroxyacetone phosphate are the plasmalogens and platelet activating factor. Both these lipids are related to the class of lipids which contain ether linkage. That is, we can say that plasmalogens and platelet activating factors are the lipids containing ether linkage. Now we see that how the synthesis of triacylglycerol is synthesized from glycerol. First of all, glycerol is esterified with a fatty acid into the 1-acyl glycerol, and the enzyme is glycerol 3-phosphate acyl transferase. This esterification occurs at the first carbon of the glycerol. Then second esterification takes place with another fatty acid or Acyl-CoA at the second position of the glycerol 3-phosphate. This reaction is catalyzed by the 1-acyl-glycerol 3-phosphate acyl transferase. Then we see that this 1-acyl-glycerol is converted to 1,2-diacylglycerol 3-phosphate, which is the phosphatidic acid. So in this way, the glycerol is converted to phosphatidic. Glycerol 3-phosphate is converted to phosphatidic acid by the action of acyl transferases. Then this phosphatidic acid is converted to diacylglycerol, is converted to 1,2-diacylglycerol, and this reaction takes place by the action of phosphatidate phosphohydrolase. This enzyme dephosphorylates the phosphate group present at the third position of the 1-phosphatidic acid to convert it into 1,2-diacylglycerol. The enzyme which catalyze this reaction simply may be called as phosphatase. Then this diacylglycerol is further esterified with another Acyl-CoA or with another fatty acid, and this 1,2-diacylglycerol is converted into Tryacylglycerol, and this reaction is catalyzed by the enzyme diacylglycerol acyl transferase. So in this way, phosphatidic acid is converted to triacylglycerol. As we see that the synthesis of triacylglycerol takes place from the glycerol 3-phosphate, and this pathway of synthesis of triacylglycerol is present in the liver and adipose tissues. In intestinal mucosa, this pathway of synthesis of triacylglycerol is called the monoacylglycerol pathway. This is somewhat different from the pathways present in liver and adipose tissues. As we know that in intestine, after digestion of food, the free fatty acids and monoacylglycerol is absorbed from the intestinal lumen into the intestinal mucosal cells. So in the intestinal mucosal cells, monoacylglycerol takes part in the synthesis of triacylglycerol. In these mucosal cells, the acyl transferases acylates the monoacylglycerol into diacylglycerol, and then monoacylglycerol is esterified, not the acylated but esterified into monoacylglycerol is esterified into diacylglycerol, and then this diacylglycerol is esterified into the triacylglycerol. So the enzymes in this pathway are the acyl transferases. So in intestinal mucosa, the synthesis of triacylglycerol takes place through the monoacylglycerol pathway. Now this diagram shows that how the triacylglycerol, which is synthesized in the liver from glycerol 3-phosphate and fatty acid, moves to blood, and then in the blood the lipoprotein lipase hydrolyzes this triacylglycerol into free fatty acids and glycerol, and then these free fatty acids are taken up by the triacylglycerol, are taken up by the adipose tissues. In the adipose tissues, again the esterification takes place uh, and so the free fatty acids and the glycerol 3 phosphate gave rise to the triacylglycerol, which is stored in the adipose tissues, and this synthesis of triacylglycerol takes place in the fed state when the um, abundant supply of fatty acids is present, while in starvation state and fasting state the vice versa takes place. That is, the triacylglycerol present in adipose tissues is again hydrolyzed to glycerol and free fatty acids, which then move through the tissues, which then moves to the tissues through the blood, where they are oxidized to provide energy, and the excess fatty acids are then again transferred to the liver, which take part in the esterification into triacylglycerol. Thank you.